Hello, and welcome to today's webinar, Current Breakthroughs in LED Cannabis Research. My name is Eric Sandy, and I am the digital editor of Cannabis Business Times. In today's event, Current's senior plant scientist, Dr. Hans Spalholz, and North Carolina State University associate professor, Dr. Ricardo Hernandez, will present results from two cannabis studies. One examines the flower yield and cannabinoid accumulation increases due to increased DLI, while the other shows how to drastically improve rooting of cuttings and transplant survival by using adequate lighting and CO2 supplementation strategies. The aim of both studies was to understand how to push the productivity of the cannabis plant further. And you, the growers in the audience today, should expect to get concrete insights from this presentation to help you improve your cultivation practices. Before we begin, just a few quick notes. You'll see at the bottom of your screen a Q&A button. And if you click that, that'll open the Q&A box. Please feel free to type your questions for Hans and Ricardo along the way. We're going to take a break halfway through after the first study, and we'll take care of some questions at that point, and then we'll have a longer Q&A at the end that will address both studies. Also know that we are recording today's presentation, and we will be sending video and audio via email to all registrants in the coming days. So with all that being said, I'm very pleased to welcome Dr. Hans Spalholz.
Thank you, Eric, and good morning, good afternoon, and good evening, wherever you may be in the world. It's my pleasure today to start off the talk on current sponsored research at NC State University with Dr. Ricardo Hernandez. And as Eric mentioned, there are two different trials that we will talk about today. The first one being essentially a DLI experiment in the greenhouse to better understand cannabis yields with different light qualities. Essentially, there's a lot of mysticism with growing cannabis, and we're trying to basically apply hard numbers to it and get more detailed information so that we better understand cannabis yields. The second trial will be on cannabis rooting, if it pops up here, and the effect of lighting and the speed of rooting establishment for transplanted plants, and then upcoming trials. So before jumping into trial one, I just want to thank research assistant Christian Collado and Dr. Hernandez for their hard work. As you can see here with our top light, they put a lot of hard work in getting this experiment set up. And now, before handing it off to Dr. Hernandez, I want to go over a few basics on cannabis and light. Most of you already know about cannabis production and the different stages, but in case you don't, there are three distinct stages. The first one is the production of plug plants, and that's anywhere from 14 to 24 days, and the plants are grown under 18-hour photo periods. Then there's the vegetative stage, and that's anywhere from 3 to 5 weeks, typically. And again, that's under a photo period of 18 hours. Then lastly, you have the flower induction, and then the growth and development of the flower product, which lasts anywhere from 5 to 10 weeks, but typically 7 to 8 weeks. So you'll see some of those terms used by Dr. Ricardo Hernandez coming up here. The next I want to go over very quickly is light. So we're all, again, familiar with hearing terms of light intensity and U-moles per meter squared per second. There's another unit. So you hear about light intensity, that's an instantaneous measurement. You also have daily light integral. So you add up all those photons, and then you get a count of photons over the course of a day. Now, to use an analogy of a rain gauge, you can catch raindrops and then measure how many inches or centimeters or millimeters of rain that you get. You can do a similar thing with photons. And essentially, you're counting and accumulating how many photons land and drop in a given spot over a certain time period. The standard is 1 day. So you get a unit like moles per meter squared per day. And why is this important to know? Because growth is correlated with DLI, or the amount of light that you get per day. And you'll hear that it's linear related. So your growth is linked to how much light you put into the system. And you'll typically hear 1% increase in light equals 1% increase in yield. Now, how does this look in the real world? So we know that through the seasons, light intensity and photoperiod changes. So you have your darker months and your lighter months. And you can see here in DLI map the different ranges of DLI that you can get within a given year. So to now focus in on one location, here's a graph for a DLI graph for one location over the course of the year. Now, you also need to know how much light your plants need. So there's no need, or there's no sense in knowing how much light you have if you don't know how much light you actually need. For example, for tomatoes and cucumbers, it's 30 to 35 moles per day. For leafy greens and young transplants, the desirable zone starts at about 13 moles per day. And again, here's a light map for a selected location of Raleigh. And on the x-axis, you have the Julian day of the calendar. So day 0 being January 1st. And then at the far end, you have the end of the year, December 31st. Then you have the amount of light, the DLI, falling on that given day. And as you assume, when you have a cloudy day, you'll have a low amount. And on a sunny day, you'll have a high amount. So this depiction here gives you a good idea of the range that you can get over the course of the year. And then this curve here is the actual average. So you have an average outside DLI of, in this instance, if you average all of those data points across the year, of about 24 moles per day. But we also know in a greenhouse, you get light depletion, as not all light makes it to the plants due to the glazing that you use, aka if it's plastic or glass, there's a permissivity value to that, whether there's dust even on those surfaces, and then you have structure that provides shading. So you always get a light reduction in your greenhouse. So now you have this data here for the course of the year, and you now know the demand of the plants, and you can basically interpret from this point where you're in a light surplus and in a light deficit. So you can see in some times of the year, you're in to the left and to the right, in case you can't see my cursor, you're in a light deficit. And those are times of the year that you need to add light to make up for, again, that deficiency. So those are some of the basics. And yeah, without further ado, I'd like to hand it over to Dr. Hernandez, and he will go over the findings of the experiments that we ran.
Thank you very much, Hans, for that introduction. Let me go back to this slide right here, because this will help us explain better the research. So the next few slides, we're going to talk about light treatments given to the plant during the vegetative stage and given to the plant during the flowering stage. Together with Current, we put up another seminar and resources to talk about the effect of the supplemental lighting specifically for the vegetative stage, specifically for the nursery industry. But today we're going to focus on the results more specifically to the flowering industry. So here's when all the treatments were applied through vegetative and through the flowering. Also notice here, this is the exact amount of time we use in each of the stages. You can see, as Hans mentioned, this can vary between 3 to 5 weeks. We use 3 weeks. And the reason for that is because the leaf area index and basically the vegetative mass of these plants were high enough for us transitioning into flowering. Also here, the flowering induction, similarly, there can be up to 8 weeks. We had to stop this experiment at 7 weeks because the majority of the flowers were mature for harvest. The photoperiod during the vegetative was 18 hours. So basically we had the sun and the lights were on for a total of 18 hours on top of the sun. And the photoperiod for the flowering stage was 12 hours. So basically we have the supplemental lighting and max total 12 hours and blackout curtain after 12 hours. Also, I want to again give credit to the PhD student that conducting all this research, Cristian Collado. He's the one that put all the hard work in the greenhouse, designed the system, collected data, analyzed, and so all the credit to him. So now let's jump to the objectives. So the first objective is to study how supplemental lighting during the vegetative and flowering stage affects the growth of the plant, mainly the flower production and quality. And the second objective is how the supplemental lighting changed during the vegetative to see if there was any impact during the reproductive stage, right? And in order to explain those treatments a little better, I'm going to use this graphic. So here in this graph, you have on your x-axis your days under the light treatment. Now the 7 weeks and 1 day is around 72 days. Here on the y-axis, you have the supplemental light intensity. In this specific graph, I give it the supplemental light intensity in instantaneous measurements, micromoles per meter squared per second. So for this specific treatment in this graph, the plants receive 150 micromoles per meter squared per second, both during the vegetative stage, 3 weeks and 1 day, and the flowering stage, which was 72. So obviously the daily light integral was different between the two stages, being lower at the flowering stage. And this is just the way we designed this specific experiment. So hopefully you have a better understanding of one treatment. So let's move on to all the treatments that we had in this research. So here again, you have 8 different treatments. Let's stay on the top line first. On the top line, you have the different supplemental lighting treatments of 150, 300, 500, and 700 micromoles per meter squared per second for the entire length of the experiment. Okay. So from lower light to high light. Now remember, this is in addition to the sun. These 700 are purely from the LED supplemental lighting. So in addition to the 700, we had the sun, and I'll show you some numbers of the sun soon. On the bottom line, you have exactly the same graphic, but now you have another 4 different treatments. The main difference between the top line and the bottom line is the amount of light intensity during the vegetative stage. For example, in this first graph, we have 150 micromoles per meter squared per second during the first 3 weeks, or maybe 4 weeks of the cycle. Mostly vegetative, a little bit of reproductive photoperiod. But then after that, the plants will move to a 700 micromoles, so around 7 weeks of the total growing cycle, they were under the high light intensity treatment. In the second graph here, you have another group of plants that the only difference between this group and this group is that during the vegetative, those first 4 weeks, they had around 300 micromoles, and then they were moved or increased the light to 700. And then you have 500 to 700, and then these group of plants are the same group of plants where they were at 700 the entire time. The goal of this is, like I said previously, is to see the impact of reducing the light during the vegetative while maintaining high light during reproductive, versus having the same light intensity through the entire time. I'm going to show you 2 videos here. The first video here shows you the greenhouse a day before they were changed to reproductive. In a different way saying it is when the photoperiod was moved from 18 hours to 12 hours. So these were the vegetative stage. That in the center is a light sensor, light intensity sensor. So we are actually measuring both the light from the LED and the light from the sun all the time. That blue sensor here, this is a temperature sensor where we're measuring the canopy temperature in every single plot. And there is a total of 12 plots for the 4 static treatments, and then 4 plots for the changing treatment. In this other video over here, this is a day before the plants were harvested to actually collect the flower biomass, a day or 2 before. And again, you can see how the light sensor has been adjusted to have accurate measurement of the environmental conditions. You can see the temperature sensors are on every single plot. You see there light given, and those light were dimmable to maintain the same light intensity from the top of the canopy to the fixture for the entire time. You can see some fans moving air. You can see cameras, so we're taking pictures every day to track the growth of these plants. And you can see the greenhouse on its entirety, right? So we have 12 plots, all distributed through the entire greenhouse gradient with the different light treatments. So very comprehensive study. So again, I mentioned again, so you have your supplemental lighting treatment, which it was at either 18 hours or 12 hours. Plus you have your natural sun, right? So here you have the sun spectrum, and then we have the LEDs on beyond the sun hours, right? For a total of 18 hours of 12 hours. So the initial light intensity in micromoles per meter square per second. Then we convert all of that up for every day to get the moles per meter square per day from the sensor in the center of the treatment. So we capture both the sun and the LED lights. And then we do the DLI accumulated, right? Because it's how many days is this the sun, the light amount per every day, sum for the entire length of the experiment, which it was 72 days. Right? So that's another way to quantify light: instantaneous and cumulative. So let's go a little bit into the results. And like I mentioned, this is specifically for the flower yield results. You're gonna see this graph here, and let me explain to you the different dots before we move forward. So here you have the instantaneous light intensity. Again, here I'm presenting the results based on the supplemental light treatment, even though the plants were affected by both the supplemental LED and the sunlight. In this graph, you have a black line, and this black line has different dots. Each dot represents one of the treatments. For example, this first dot, but this image here represents the static treatment that had 150 micromoles for the entire length of the growing cycle. And this represents the yield, and this is the flower being dry, a dry mass of the flower, right? This now blue dots, every time you see a blue dot, is a dynamic treatment when the light intensity changes between the vegetative and the reproductive. For this specific blue circle, we have 150 in vegetative and 700 in reproductive. So black and blue, static, dynamic. So let's remove that for a second. So, let me go back. So basically right here, you can see that 150 micromoles had a lower, for the entire time, had a lower yield than growing any vegetative from 150. Remember, this is only 3 to 4 weeks, and moved to 700, right? And to me, this is obvious, right? Of course, more light, more yield, right? Now here, similar. We have the blue line, the blue dot, high variability in the plants, and we have a 300 light intensity the entire time, or 300, then move to 700. Again, more yield at the end, flower mass, because of the higher light. Here again, we have 500 and 500 and 700. Again, more yield if you have higher light. And at the end, we have the final point, which is shared between the two treatments, which is 700 the entire time. So basically here you have 150 the entire time, 300 the entire time, 500 the entire time, 700 the entire time. Here you have 150 for the vegetative, move to 700. 300, move to 700. 500, move to 700, and 700 the entire time. So if I show you this image, it's not very clear what's the impact yet. So in order to make this more clear, instead of presenting the results instantaneously, right, which is this line here, we're going to convert those numbers to present the results cumulative, right? How much photons of light fell on the plant the entire 72 days, independently where you apply them, either in the vegetative or in the growing. So this is this graph over here, So now it doesn't matter when you apply the photons. With this graph, is just telling the total amount of photons received by a plant for the 72 days, which is moles per meter square. So if the plant was under 150 the entire time, then it was 150 micromoles every second, times how many seconds on a day, and then times the number of days, right? So this is this much light, around over 1,021 moles per meter square for the 72 days. This one is a 300 the entire time, right? So that will lead to around 1,556 moles of light for the entire 72 days. Now you have two different shades of color. You have the gray and the black. So with the gray ones are the ones that were dynamic. So for example, this one right here, it was 150 micromoles during the vegetative, moved to 700 during the reproductive. And these ones right here is this almost the same amount of light, right? Because being a 500 the entire time, or moving from 300 to 700, depending on vegetative and flowering, it gives you almost the same amount of total micromoles of light, moles of light. And then you have 500, 700, and 700 to 700. So two things to notice here. The first one is that it didn't matter that you give 700 micromoles of supplemental lighting, plus 17% more from the sun. The increase of flower dry mass was linear compared to the increase in light. How predictive is this? Well, I can tell you that based on this research, we get 0.21 grams of dry mass per every mole of light added into your growing system. And here it was independently if it was given by LED or the sun, right? Now here you can see that the lowest amount of yield is also the lowest amount of light, and it's around 50-50, 50 from the sun, 50 from the LED. And the highest amount, it was most of the total amount of light here of the almost 3,000 moles per meter square, around 83% were from the LED and 17% from the sun. All right, so number one, it was a linear response. We never got to saturate based on these light levels. The second thing to notice in this graph is that it didn't matter if you gave them dynamically or if you gave them statically for the entire amount. What mattered the most is the total amount of light. That's why these two lines in the same dot, and this lands exactly where it belongs in the line based on the cumulative. And these lines almost exactly where it belongs in the line based on the cumulative. Another way to put that is like, if you reduce the light intensity during the vegetative, most likely you will have a penalty during your flowering. If you're able to keep the same high light intensity during both vegetative and flowering, you will have a better yield if you reduce the light intensity for vegetative. Because this graph is telling us that it's cumulative, right? So do not dim those lights during the 3 weeks, 4 weeks vegetative, because you will pay the price at the end. So these are the plants look. These are the ones from static treatments, have 150 all the way to 700 micromoles per meter square per second. Plus the sun, you see the difference in size, very obvious. Here you have the plants move from dynamic 150 to 700, and this plant 700 is the same, right? Here you have a pictures where you can see both, right? This is the plant, one plant at the end. You remember these plants were grown as a community, so you can see very high amount of leaf area and foliage here. We have data on biomass and partitioning and all of that, but we can talk about that on a different time. Right now, I want to talk about what's inside those flowers. So this graph right here is my attempt to show you that if I'm able to increase the CBD percent on that plant, that will directly increase the THC percent of that same plant. This is actual data that Christian collected and analyzed in the lab to show us that if we drive CBD, we're going to drive THC. Therefore, even though this cultivar was Cannabis sativa, but mainly a CBD producing plant, because we're in a university, we need to work on hemp CBD plants. These results can apply to a THC producing plant because the relationship is 1-to-1. Okay. So it's good for both markets. So after making that clear, now let me show you this graph here. Now on the x-axis, again, this is the cumulative amount of light through the entire 72 days, right? So the greater the light, the greater the yield. You have two shades. Again, you have the gray shade, which are the dynamic changes, and you have the black shade, which are the static light intensity. So for example, this was grown under 150 the entire time. This one's under 300 the entire time. This one was grown 150 to the vegetative, then moved to 700 during the flowering, and so on and so forth. Then you see two shapes. The first shape is a diamond, and the diamond shows you the total cannabinoid content. And then you have your triangle, which is only the total CBD content, right? And the cannabinoids are made of the list over here on your side. So here you can see your total CBD and cannabinoid percentage depending on the diamond or the triangle. And then you don't see any lines drawn in here because we found that the increase of light intensity did not affect the percent CBD per 1 gram basis, right? So the concentration was the same, right? More light didn't reduce the concentration. Less light didn't reduce the concentration. Basically, you have 1 gram of flower. That gram of flower had around between 18 and 20% cannabinoid content, right? So it's important to see increase of light intensity did not reduce that concentration. However, if we plot this slightly different, how about your total yield, your total potential revenue, right? So in this graph, now you have, again, your total amount of light accumulated for the entire amount of time. Again, you have two shapes, the diamond and the triangle. The diamond is your cannabinoids, the triangle is your CBD. And then you have two colors, the gray and the black. The gray is the ones that moved from lower to higher, and the black is the ones that were static, right? So this is your 150, 300, 500, 700. This one moved from 150 to 700. This one moved from 300 to 700, and this one moved from 500 to 700. So you can see now that the increase in light increase your total CBD and cannabinoid yield in grams per meter square linearly to the increase in light. So basically, in this example, it's telling me that if I added more light to these plants, they got more yield. And based on the light levels we were using, around 3,000 moles for the entire 72 days, we didn't see a saturation of that response. Right? I'm sure it's there. We just didn't capture it here at these light levels. So now let's put that into perspective, right? So here you have the same graph, but instead of your grams of CBD per meter square, I converted that into potential revenue for two different crops, either CBD flower or THC flower. And you can plot this based on current prices of CBD and THC flower, which are a little bit different than this. They continue to change every day. But for this specific example, you can see that the increase of light increases the revenue. Now, here I'm also assuming only half of the flowers are commercially viable. Me, as a scientist, I wanted to use all the flowers to make commercially viable, but talking to some colleagues in the industry, they say, no, every flower is commercially viable. And if it's commercially viable, it's a different price category. So I say, okay, I'm gonna assume 50% of the flowers are on the smokable flower price category, and the other 50 we can put it somewhere else. But this is just thinking about that 50% of viable sellable flowers in the highest smokable flower category, right? So based on that, we see the revenue based on the yield. So basically, I took these grams per meter square and multiply it by the value, and then we have for every meter square of growing area, I can make $177 when growing plants at 150 micromoles for the entire time. Or I can make $740 if I grow the plants and the 700 micromoles plus the sun, for the entire time. Right? Now, if I'm selling mainly CBD flower, it's a lot lower, but it's still very profitable. So at around $33 per meter square of greenhouse environment, or around $136 per meter square in the 700 or 3,000 moles of light. But well, how much it costs to keep those lights power? Well, that depends on the cost of electricity. It also depends on how long are the lights on. It also depends how efficient the fixture is. That's why it's important to pick a fixture that is very efficient, so it cost is lower. But in this specific example, this is the same graph that before, but now it's showing you the yields, right? This is the potential revenue. If I add another line, that's the cost of electricity to power a light this specific efficiency. It will cost me $25 for every meter square. $25 for every meter square to light the greenhouse at 700 micromoles per meter square per second using this specific light efficiency. Right? So that difference in revenue, it's going to cost me 25, but I can make 136. It's going to cost me 25. I'm going to make $740. Now here you have to reach out to Hans and say, well, how much is going to cost me to actually outfit the system with all these lamps, and what will be my return on investment? That's beyond me. But this gives you an idea of where the investment on the light based on running the light at different levels. To summarize, so a couple of summary points, and then we'll make a quick pause for any questions. So for this specific cultivar, 1 mole of light produce 0.2 grams of dry flower per every meter square area, right? 1 mole of light, right? So you have more light, you have 3,000 mole of lights, you will have the grams that we use for these specific calculations. The potential revenue is production minus electricity costs, right? So for CBD dry flower, and these numbers can change, and you can change it depending on what you're getting for your flowers, can range between $2 to $100 per meter square, depending on the light intensity. For THC flower, again, these numbers can change. It may be $160 to $700 per meter square, depending on light intensity. And then in order to make these calculations, these are the changes you can make in your calculations. Is as the assumptions were that 50% of the flowers are market viable. All light is sole source. CBD flower market rate is around $498. That has changed, so you can change that number. THC market rate is around $2,000 for the smokable flower, $2,600. You can change that too. The fixture efficacy is 3.1 micromoles per joule. That's the efficacy. That's pretty high. In LEDs right now. That was the efficacy of these lamps that we use. I also use the average electricity cost rate. This is 9 cents per kilowatt hour. So you can change that if you're paying more than that. Most likely you're not paying less, but you may be paying more. And I didn't include any of the production costs because I figured it will be standard based on practices. So I'm going to stop right there to see if there's any questions about this specific study, and then we can move to the next trial.
Thanks so much, Dr. Hernandez. That was a great overview of that study. We do have a few questions here. And again, I'll just remind the audience that we have a Q&A button at the bottom of the screen. We'll hit a few now, and we'll certainly circle back at the end of the hour. Just real quick, as a point of clarification, one audience member was asking if that term, this was on the y-axis in a few graphs, if that term flower dry mass is a true dry mass, or if that includes the industry standard of around 12% moisture content.
Great question. That includes 12% moisture content.
Okay.
We actually follow the guidance from some of our colleagues to see how we typically bone dry our biomass for other research, but this one is the industry standard.
Okay. We had a couple questions on cost there on one of the recent slides. One audience member was asking what the costs are using kilowatt hours as the metric. Are you able to break it down that way?
Well, not on the spot, but it's very easy here. So you have 9 cents per kilowatt hour electricity, right? So basically, you have this lamp on, well, you get the wattage of the lamp, right? So we did a light plan. We find out how many lamps we need in a commercial facility, right? Not research, this was commercial facility. And then we calculated the wattage of the lamp at the time the lamps were on. But if you want to do a back of the envelope calculation, if your electricity cost, you can do that, divide the cost by the kilowatt hour 9 cents. So if we go back right here, and I won't spend a lot of time on this so we can get other questions, but you can get these costs here, $25 per meter square, and then 9 cents, right, per kilowatt hour. So basically, I need to give you how many kilowatt hours per meter square. So yeah, you won't be able to do it. So we can create that graphic. We can create that graphic and add it to the slides. Yes.
Excellent. Yeah, we can certainly make connections after the webinar here. We have another kind of formula related question. One audience member was asking if you might be able to provide a formula for using, let's say, 1,000 LED light units, for plants per lamp. I guess taking the formula that you were just referencing in that recent slide, what's an easy way of extrapolating that for 1,000 light units?
Yeah. This is a very good question, but You're not gonna like my answer. Haha. But anyways, I gave it to you. Oh. So basically this formula right here, let me show you. It's we're assuming a few things, right? So the best way to get what you need is to contact a light provider, horticultural light provider, and tell them to make you a light plan, right? You give them your area, and then they can give you a light plan. You didn't how much light intensity you want. And based on light plan, then you can calculate how much energy you're gonna use based on how many lights you have. That's the best simple way. Most companies are very happy to provide you with a light plan, right? I know GE is, Current is. But the way I did it is using some assumption, right? So I took the light intensity, the desired light intensity, the area grown for a big greenhouse. I got the fixture efficiency. Remember, those current lights are around 3 micromoles per joule and higher. So use that one. Took the power of the lamp. And then here's when it gets tricky. There's utilization factor and maintenance factor. Utilization factor is how much of that light you assume is going to fall in the plant. If you have a very large greenhouse and you have good distance between lamp and plants, you can make an assumption that maybe 90% of the photons will fall in the plant and 10% will escape the system. But you have a smaller house and some of the photons are escaping, then that factor will be a lot lower, right? It can be more like 50, 60%, which you're wasting a lot of photons. So that's, I assume here 90% of the light is being captured, assuming it was a light manufacturing, a large greenhouse. The main factor is how long the LED lights under that light intensity. And then based on that, I got the calculation of how many lamps I need. You can use this, you can plug all your numbers here, use a utilization factor that you think is obvious for you, 50%, 60%, 90%. Maintenance factors just use 90%. So you can use here 0. Whatever you think how many photos are capturing by lamp, and here you can just 0.9, and everything else you can find it, and you will give you how many lamps you need per meter square or per a specific area. But you probably aren't gonna undershoot or overshoot. So light plans are useful for that.
Excellent. Yeah, and of course, as a reminder to the audience, we'll be sending these slides around to everyone. The video will be sent via email, so you'll have this to reference. One last question before we go on to the next study. I want to be mindful of the clock here. It's a question about wavelength. Was the wavelength of the lights in that first study between 400 to 700, or could you clarify that?
Yes, all the plants were under the same light intensity and also, same light spectrum. And I think this specific fixture, Hans, correct me if I'm wrong, it was close to 18% blue, 20, and the rest being red, right? So around 20, 80% in that area. Now we're getting ready to do another study specifically on spectrum. We use this light because it's high efficiency. So we wanted to show, high efficient light. So we work in other lights that will have different colors. So that's research right now. We'll start soon in my lab. Yeah. So around 2080, I think, Hans, if that's correct.
Correct. Yeah.
Excellent. Well, I'm going to make note of a few questions here that maybe I'll keep in my back pocket for the end, but I do want to, we can move on to the second study here, and I'll hand the mic back to you guys. Thanks.
Thank you very much. So moving on to the second study, now we talk about the plants in the greenhouse. We talk about the vegetative impact in changing the light and all of that. Right now, we're going to talk about effects of light on rooting of cuttings and its correlation with establishment of transplants. So basically, there is a whole market, nursery market, that works specifically in going the starts, right? So you have cuttings, you have the rooted cuttings, and then this make a new crop, right? So this is a nursery. So a very important step here is to get those plants to root. We want to convert lots of those plants, not only want to convert high percentage of those plants, those cuts, but also we want those cuts to have very healthy root system so they can go into the growing environment and produce great plants. So in order to do that, we did some research to look at the impact of the environment under the rooting of these plants. And again, my PhD student, Cristian Collado, was the main soldier and worker doing all these experiments. So he deserves all the credit. but we're going to look at the impact of light intensity mainly on these plants. Why is this important? Well, this is an important market too, right? So here you have, for specifically for hemp, now if you are a recreational grower for recreational cannabis, you have your own prices, and they may be around close to hemp now, or close to hemp in 2019. But anyways, one clone here, the average price for a cannabis clone, rooted clone, is around $4.47. But that can range between $2 and $8 depending on the market, right? How mature is the market? If you're in a mature market like California, you may be close to the $4 price point. If you're in a new market like Michigan and others, you may be close to the $10, $20 per plant. You know your prices. This is just an example. So let's say you can make around $4 per clone on a medium market, right? Now, how does that compare to other plants, to other horticultural products? Well, 1 kilogram of tomatoes, right? It will give you around $2 per 1 kilogram of tomato. And based on tomato yields, right? Based on tomato yields found in the literature, that will make you around $60 to $100 per meter square of greenhouse area. All right, so nice, profitable. That's why we grow tomatoes in greenhouses. Lettuce. Now, lettuce is a little more expensive than tomato. All right, and also lettuce. Remember, the majority of the lettuce actually is part of the product, right? The whole head, all the leaves are your final product. While the tomato is the product, but the whole plant goes to compost. So here the lettuce, is also sold between $6 to $9 per kilogram of lettuce. So that'll give you around $269 per 1 kilogram, up for 1 meter square of greenhouses. Grafted tomato plants is a high value, 1 plug, maybe $1.20. So that'll give you around $500 per meter square. Hemp plugs can be plugs. If I am assuming $4 per unit, you can change it to whatever you have right now. That will give you around $1,400 to $2,100 per meter square revenue on a nursery that is rooting plants. So a lot of value of this nursery industry. So why plugs from cuttings instead from seed? So we know it's mainly to maintain the genetic uniformity and therefore the chemical uniformity of the plant. We want those CBD and THC yields to be where they're supposed to be. However, there's a lot of effort on seeds and feminized seeds, and that's also valid, right? But this specific research is on the cuttings. Right? So objectives, well, we wanted to see the root growth of Cannabis sativa and the different light intensities during the rooting environment. And the hypothesis is that high light intensities for propagation of these plants may cause stress and lower root development, right? When you have too much light for a plant that no roots to begin with, that may be a stress. And we know that because we understand that from the ornamental market, the cuttings from the ornamental market. We understand that from grafted plants. You have too much light when the plants are healing or rooting, that affects the rooting. That's actually a negative impact than positive impact. So Christian designed a few light intensity from 50 to 400. For rooting cuttings, typically is between 50 and 100 rooting cuttings from ornamental plants. So we went to levels very, very, very high levels here. So we measure the light intensity for every single plant, and we use 50 cell tray plants, and then we did 12 cuttings per treatment. Right? It's a quick video of. Nope, it's just a picture. I'm sorry, I thought it was a video. It's a picture of the healing environment. Not very good picture. So we took cuttings. We tried to standardize the cuttings for the experiment. We went from mother plants and different cuttings with different fresh mass and different height, but they were standardized to have a similar response. So the height was around 4 inches, 2 fully expanded leaves. The fresh mass vary, the stem diameter vary, but we actually collected all that data. This is the information for the rooting environment. You will have this recording, so you can look at this and compare it to your own rooting environment. This is the rooting environment we use for this research. Light intensity, like I said, there were multiple light intensities from 50 to 400 or close to 400. This is a light spectrum. This is your CO2 concentration, ventilation rate, and so on and so forth. So let's move into the results. So this is the results of a cannabis plant rooted for 14 days in this chamber, right? So here on your x-axis, you have your light intensity in micromoles per meter squared per second. And then on your y-axis, you have your root dry mass, right? So the higher the dry mass, the better. So you can see here in the center, the increase of light intensity. So since we measure light intensity on every plant, the increase of light intensity increase the biomass, root biomass growth up to around 300, and then it flattened, right? So the root biomass was around 0.01 grams for the low 50 light intensity and around 0.065 grams on the highest light intensity before saturation. Now here I was very surprised because I was going to think that saturation was going to be somewhere around 100 and then have a flat line. But no, Christian found out that actually these plants can take a lot more light when rooting if you're controlling the humidity very well. So I was very, very surprised about this. And not only the roots, right? It also affects the plant shoot dry mass, and plant fresh mass, right? The increase of light intensity increase the biomass of the top part, not only the roots, but the top part. But the rate of change was a lot higher in the roots. So again, this is the same graph. I just change a little bit and you have it, the increase of light intensity flattened around 300, but everywhere increase the root biomass. But we also did this under a higher CO2 level. So basically we shift the root dry mass development by the increase of CO2. All right, so not only the light increased root, the CO2 increased root also. So we were very surprised by the response of these plants. So what was the point of that? Yeah, a plant with more roots, what's the point? It's a nice plant. Well, number one, it's a nice plant that you can sell as a propagator, but as a consumer of this plant, you want to see if there is a long-term effect. We didn't have a lot of time to carry out the long-term effects of the plant, but we did potted several of the plants and grow them for additional 14 days in the greenhouse under the same environment for all the plants, right? So these are the plants moved to the greenhouse to measure their growth for another 14 days. And what Christian found is that yield, that the biomass of the plant was maintained after 14 days. So basically, under the same conditions, 14 days later, the plants that were rooted under higher light intensity have higher plant biomass. 53% greater between the ones on the highest light intensity and the lowest light intensity. Now, will that carry to the flower? We haven't done that research, but it carry through, it can carry through the vegetative. And since we know now that vegetative growth impact has a huge impact on the flower, this may be related to the flower also. So summary, we have the increase between 50 and 300 was a 6-fold increase in root biomass. We didn't see a root biomass increase above 300, so maybe that was saturated. Now you need to understand that this was under the chamber that the humidity was very tightly controlled as a sensor. If you try to do this in domes, you may have a challenge because there may be a lot of stress since the dome is a passive control of humidity. So you have to have very good control of humidity. It was after 14 days in the greenhouse, we saw also an increase in growth biomass in the greenhouse. And also, just an observation, 100% of the plants were rooted and survived in the greenhouse in these light treatments between 150 and 400. And only 83% of the plants survived the greenhouse transplanting conditions between 50 and 100. So we tend to do other research. We want to do the spectrum, different spectrums from blue and red wavelengths and white wavelengths and some far red wavelengths. We also going to do, so this will start here this November. We actually are fitting the greenhouse right now. And this is what I have, and I'm going to open the floor for some questions. Thank you very much.
Excellent. Thank you, Dr. Hernandez. And yeah, we'll certainly point folks to the contact information there on the screen, as well as the booth location out at MJ BizCon in the near future here. We do have a couple questions. I want to pick up the thread from the first study, and we actually got a couple versions of this question. Dr. Hernandez, is there a point of diminishing returns when raising light levels? On one hand, I guess to play the devil's advocate almost, why not increase the DLI well past 700?
Yeah, good question. So let me move to that graph that I want to show you here for that answer. Let's do this way. So yes, there will be a point where there is saturation. This may also be related to type of cultivar, but it would certainly be related to the way you handle water. These plants and the different light levels, they have different irrigation schemes, right? So more light, those plants were using more water. So they were being watered more. The way we did that is by sensor and weight. Christian installed some scales and was measuring the water usage, so they were being maintained at the same moisture content in the substrate. If you go ahead and increase the light intensity without changing your management conditions, such as water and nutrition, then you may not see that yield increase. And then you won't see that return on investment on your lights. So it's important to adjust the way you grow plants when you adjust in the light level. Most likely, yes, there is a saturation point. For this specific plant, at this specific light intensity through the entire period, we didn't get to that saturation point. I was almost sure we were going to get there, but we didn't. So it means for this specific cultivar, we can add more light as long as we manage the watering and the nutrition with the water accurately based on light demand.
Excellent. Yeah, and looking at those different treatments, I see, of course, the 7 different treatments there on the screen. Did all those receive the same VPD and
yes.
CO2 levels?
Yes,
okay.
They were all normalized to the same. Yeah, they were all in the same greenhouse, and we had a sensor everywhere. So if there was a temperature change, because you're in a greenhouse, there's temperature gradient, we adjusted that because we knew it. We knew it for every single plot. So we used that data to analyze the results.
Excellent. Moving back to the rooting study, could you, what were the humidity levels at the different days of rooting in that second study?
So for this specific study, this is not optimized. Okay, this was not a study for humidity. Oh, I see why you mentioned that. It's not here, huh? Yeah, right here. So percent humidity was maintained around 99.8 for all the study length from beginning to end. Like I said, that's not optimized. We've been doing trials with that and doing dynamically BPD control to maximize rooting and plant growth, right? I know you don't need to have this humidity the entire time. We had it here because we wanted to just test the light impact. Now we're working on dynamically changing the humidity together with the light impact. But it was maintained very high, yeah.
Excellent. Does the overall flower quality change or diminish or increase as the overall flower biomass increases? I know this was something you were addressing earlier. I suppose that word quality could mean several different things, but does the overall flower quality change as that biomass is increasing?
Not that we can measure. Let me rephrase that. I don't think we the percent CBD, percent THC did not change, right? So that was stable across it. Based on mold development, all that wasn't any difference, but that was more related to the cultural management. Now, I don't think we're not experienced enough to know if the smell, we measured the terpene, there was no difference in terpene concentrations, but other than that, based on terpenes and based on cannabinoids and based on the way the flower looks like, and based on analysis of microbes, no difference. If there's something else, we don't know, right? Like the smell or we don't know. We have to get someone in there to tell us. Yeah.
Excellent. Well, I know you had ended on that slide with some contact information and some connections beyond this webinar, but I did want to sort of combine a couple questions here for Dr. Spalholz in terms of potentially next steps or action items that folks can take if they want to continue this conversation with Current or even begin perhaps an onboarding process of some sort.
Sure. Yeah. You know, this is really good research and we're always excited to talk about it, as well as the upcoming research as well that's in the works here. But yeah, we'll be at MJ BizCon upcoming here in November, and please stop by the booth. You can see it on the screen, 1251. And yeah, we'll have a good conversation. We'll have myself there, plant scientist, and then other engineers that can talk to some of the questions that you have on electrical usage, CapEx, OpEx, all of that.
Excellent. Sounds good. Well, I know, the questions from the audience were fantastic. I want to thank everybody in the audience for attending today, as well as Dr. Ricardo Hernandez and, of course, Christian Collado from NC State for the research there. And I do want to thank Dr. Hans Spalholz and the whole team at Current for providing this presentation. As a reminder to the audience, you'll be getting a link to the archived video and audio. That includes all the slides, of course. That'll be coming via email in the coming days. And we do have a couple folks who left their own contact information. We'll make sure that gets to the team at Current. But again, Dr. Hernandez and Dr. Spalholz, thank you so much.
Thank you.